description of my own first summer internship when I was a high school student working at the University of Kansas. This is Sydney Cameron, who's currently a professor at the University of Illinois at Urbana-Champaign. When I first got to know her, she was a PhD student at the University of Kansas in the entomology department studying bumblebees. That's the bees. This is what a typical bumblebee nest looks like, and in the nest you have a bunch of bees. You have one queen bee, which is the only one who's laying the eggs, and the queen bee maintains order in the hive and keeps everybody else from laying eggs. And that's what we were really studying, is how this order is maintained. And specifically what happens if you remove the queen, the other bees notice that, and they sort of establish a new dominance hierarchy, and one of them starts laying eggs. So this is what it looks like when you open a bumblebee nest, but in actuality, it looks more like this inside a bumblebee nest. They're typically underground, and it's dark. So the way the bees communicate is by touching each other, and buzzing, and most importantly, chemical communication. And so the way the queen tells the other bees that she's the queen and she's in the nest is she releases queen pheromones, and then the other bees recognize those and they don't lay eggs. What happens then is if you remove the queen, this is the abdomen of a bee. The queen bee has very large developed ovaries and lays eggs. A normal worker, the ovaries are underdeveloped. When you remove the queen, one of the workers starts developing its eggs, developing ovaries and laying eggs. And so we were interested in determining how the bees decide who's going to be the boss worker, and the other ones are kept from laying eggs. So when you have a bumblebee nest like this, the tricky part is if you remove the queen, you have all these other workers, and you need to decide who's going to be the boss worker. And these bees, at least to human eyes, all look the same. And so what you do is you hire a high school student who sticks a number tag on each bee, and then you watch the bees, and you can say, oh, bee number 47 is starting to be dominant, bossing the other bees around, and then eventually maybe you can see bee number 47 laying eggs also. And in addition to what we did then is, the order is maintained also by pheromones, and bumblebees, like other bees, produce a lot of different pheromones, and specifically the mandibular glands of the queen produce what's called the queen pheromone. And so the other part of my job was to reach into the bumblebee nest, pull out individual bees, have them bind into filter paper, and then we would analyze that by mass spectrometry and could show over time then that the worker that was becoming dominant not was only physically dominant, but also was developing queen-like pheromones, and that way was suppressing egg laying by the other workers. So as you can imagine, there's a certain amount of risk involved in tagging 100 bumblebees and pulling out individual bees from a nest to let them bite in filter paper, so I got probably stung well over 100 times that summer. But nevertheless, I continued in research, and probably this is the start of my research career. I hope your summer internship was nearly as painful as mine, and that you had a good time, and that you will also continue and go on into research careers. With that, I'd like to start by introducing our judges. We have Fei Wei Li, who's here. We have Linnell Randolph. We have Lena Miller. We have Adrian Powell and Alexa Schmitz. This is a friendly competition. They'll be listening. They may ask questions at the end of your talk, but otherwise, just be aware if you want to talk to somebody in particular, rather than one of your friends, maybe talk to one of the judges. I'll be keeping time, and each of you has 15 minutes. We'd like to finish by about 12, so we have some time for questions. So if I hold up this sign here in the front row, you'll see it hopefully, and maybe think about wrapping up your talk. With that, unless anybody has procedural questions, I would go ahead and introduce our first speaker, who's Arianna Nimocks, who's working in my lab this summer together with Malcolm Boldemariam, and has been looking at insect defense in caterpillars in maize.
Good morning. My name is Arianna Nimocks, and I wanted to thank you, first of all, for coming out here today. I know it's early, but you got your breakfast in, so you should be up and ready to learn about maize defense. So my project title is Determining the Functions of Herbivore-Inducible Maize Genes in Defense Against Phytophthora exigua. And first I'll start by talking about what jasmonic acid is, or JA. JA is a plant hormone coordinating both development and defense against herbivore attack. So this will include two main ways. You have direct defense, which is basically hurting the bug that's feeding on your maize plant. And then you have indirect defense, where you're actually attracting the predator of the herbivore that's feeding on you, say a wasp, so it can go in and eat the caterpillar. And then, so now I'm going to go into the biosynthesis pathway of JA. And that starts off with a little molecule called alpha-linolenic acid in the chloroplast membrane. That's going to go through a series of enzymatic reactions, and it'll eventually become JA. JA is then turned into its bioactive form, J-isoleucine, by an enzyme called JAR. What JAR is going to do is conjugate JA to isoleucine, the amino acid. Once you have that form, then that J-isoleucine, represented here by the red dots, is going to attach the repressor, JAZ, to something called the SCF-CoI complex. And what that's going to do is it's going to target JAZ for degradation. After that, JAZ is going to be degraded, which means that you have thousands of target genes being turned on. And what's important to remember here is that these genes are not made just for the fun of it. They're not translated just for the fun of it. A plant really has to garner all of its resources and energy into these metabolites that are then going to be translated. So these are very important in defense, and that's important to remember for later. Now, my project is twofold. I actually did two projects this summer, and they're focused on two genes that are highly upregulated after herbivory. One is called IA Amino Synthase. And the reason why it's a very interesting gene is because of two main reasons. Firstly, it's highly upregulated after herbivory. So that's really unusual because, as you can see, it has no place in the JA signaling pathway so far. So what makes it so highly upregulated? And then the second reason why it's so interesting is because it's actually related to this enzyme called JAR. And that kind of makes sense because IAA amino synthase, or auxin amino synthase, adds amino acids to auxin. So what if it can add isoleucine to JA? That's what we're focusing on. Then the second project is focused on the gene ZMAOC2. The reason why we're looking at this is because all the spotlight has been in dicot so far for JA biosynthesis. So there isn't too much known about the function. Actually, it hasn't even been functionally characterized in maize. So we're trying to find out how similar monocots and dicots are. Then I'll go into why maize. So why are you sitting here for a 15-minute lecture hearing me out about corn? So the reason why we're studying maize is because, firstly, it's economically very important. So just in 2014, over 1 billion tons of corn were produced. And unfortunately, there's a lot of stresses that the maize plant has to defend itself against, including caterpillars and aphids. And this means that when there's an infestation, the farmers have lower yield, obviously. The second reason is genetic variability. You have a lot of genetic variability between different strains of corn, so that makes it easy to kind of play with scientifically. And the third reason is, like I said, all the spotlight has been on dicots, but we're trying to figure out more about the JA pathway in monocots. Okay, so I'll go into my first project, all the methods and procedures, and then the results, and then I'll go into my second project. Like I said, so this is actually the phylogenetic tree that shows IAA amino synthases right here, and how they're related to the JAR enzyme. So we're trying to figure out if IAA amino synthase is basically a JAR enzyme. So overall methods and procedures, we use something called transposon-based reverse genetics. And you start with a mutated genotype, which we did genotype, and you check all of the phenotypic effects. So you'll check silencing efficiency, caterpillar performance, untargeted metabolomics, and phytohormone analysis. And I'll go into each of these in depth. So the first thing we did was we had to do the silencing efficiency or gene expression knockdown. You start with RNA extraction, which many of you probably did, and then you get the cDNA, you run that through QRT-PCR, and what we got were promising results. So you have lower amounts of the IAA amino synthase gene in the mutants compared to the wild type. So this is kind of like a preliminary stage to all of the later investigations that we're going to conduct. After that, we did caterpillar performance. So that included putting about 300 caterpillars individually onto maize plants, and then weighing them after 5 days of eating. And the basic gist of this is that if you have caterpillars with higher masses, that means that the plant is less defensive. The caterpillar likes the plant better. And what we saw was, after using a two-factor ANOVA, a significant increase in one of the genotypes for caterpillar mass. So that's really good results. So now what we did was we tried saying, okay, caterpillars, you liked this maize plant. What chemicals are in it so that you liked it so much? Why did you like it so much? And what we found after doing something called untargeted metabolomics, which is looking at all of the chemicals in the plant, is there we go. Okay, is that you have clustering between. Oh, there's the little pinwheel. Okay, that's okay. Well, I'll continue. But what you have is clustering between the different genotypes. So all of the colored dots that you see, those are mutants. And the mutants are clustered away from the wild type, which is in black. And that means that there's a difference between these two genotypes. You might ask, why is there clustering? Well, we showed using this heat map that there's a significant difference between the wild type, the chemicals in the wild type versus the chemicals in the mutant lines. On the top here, you have the genotypes. Black is wild type. Well, if you can remember it in your head. So black is wild type, and then the colored versions are mutants. And you have a sharp divide, if you can remember, between the mutant lines and the wild type. So that means that there is a significant difference between wild type and mutant lines. So after this, what we did was phytohormone analysis. So now we're going even deeper, and we're saying, what about the JA and JA isoleucine concentrations in these plants? And what we found was that there was a lower amount of JA in the mutant lines compared to the wild type lines. And it has a significant difference. Same with the JA isoleucine, which is critical because we believe that JAR, or that IA amino synthase, makes JA isoleucine. So you do have a lower amount of JA isoleucine in the mutants. So I'll just go ahead. After this, what we did is now we're going to transition into the ZM-AOC2 story. So this is just kind of a refresher, but we're looking at the function of ZM-AOC2. And we start off this section of the project with the same kind of pipeline. So you have caterpillar performance. We're telling the caterpillars, have at these plants. Tell us which plant you like best. And from that, we can know which plant is the most defensive. So after caterpillar performance, and this slide kind of shows, again, the preliminary. You have genotyping on this side. So we did find lower transcripts in one of them, but there was no significant difference. But for the caterpillar performance assay, we found that one of the mutants had a higher average caterpillar mass, which again is good, but that this one is significantly lower, and that's kind of unusual. So we're going to have to do more replicates on that later on. Then again, we did untargeted metabolomics, so digging kind of deeper into the plant. And what we found was that there was significant clustering. Again, if you can kind of remember the cluster map, there is significant clustering between the mutants compared to the wild type plants. And the heat map will show, again, just imagine, that there is a significant divide between the mutants compared to the wild type. And after this, we did phytohormone extraction looking at JA and JA isoleucine levels. And on average, we did find a lower amount of JA and JA isoleucine in the mutant versions. It wasn't significantly different, but we're going to do some more tests on that and probably more replicates. So the conclusions overall are that IAA amino synthase does have an effect in JA flux because of the experiments in caterpillar performance, in untargeted metabolomics, as well as the phytohormone extraction. We were able to find that IAA amino synthase may have a big role in JA flux. For ZM-AOC2, we're still going to have to do some more work on replications, but overall, we did find some good results, notably that there were lower average JA and JA isoleucine levels in the mutants. And then this is kind of, so further experimentation. If I had 12 weeks in the REU, what we would do is we're going to clone the IAA amino synthase protein, and then we're actually going to add JA and isoleucine into a mix. And voila, if we get JA isoleucine, that's a really good indication that IAA amino synthase can conjugate the two together. So that's kind of, like I said, future research. If I can, I just want to show the pictures of the acknowledgements. Well, I think actually most of the people are in this room actually. So I wanted to thank my PI, George Jander, and all of the Jander lab for letting me use their lab for 10 weeks. And also a special thank you to Mel Cameron, my mentor, who's been great in showing me the A to Z of this whole experiment. And I also wanted to thank my funders, so NSF, BTI, and Cornell for providing me with a good stipend and food. Thank you.
We got time for questions?
Oh, you're right.
Questions?
Yeah, you said you wanted to do more replicates. How many replicates did you actually have?
Yeah, so For the caterpillars, we had about 300 caterpillars in total, so that means, I believe we had 40 replicates per genotype. For a couple of them, we would like to do some more. We did have a couple that didn't have the transposon insertion, so we only did fewer, we didn't do that a second replicate time, so that one only had about 20 replicates. Yes?
So you had mentioned that you place these caterpillars onto these plants and then weigh them to see what, to test, I guess, plant defense.
Mm-hmm.
But how do you go about knowing that the results that you're getting are solely based on plant defense and not based on caterpillar resistance to those defense chemicals?
Okay, so that's a good question. So part of the reason that we did the untargeted metabolomics and phytohormone extraction was to make sure if it was because of defense chemicals. So using those two methods, we're kind of cross-checking to see if the plants that had higher caterpillar mass actually do have more defense chemicals. So that's kind of our cross-checking that. I don't know if that answers the question. Anything else? Yes.
So how do you know that the caterpillar mass is in direct correlation to like the amount of defense the plants have versus something like, I don't know, what the caterpillar's genetics are or something to do with the caterpillar itself?
Okay. Let me think. Again, so part of it is kind of using the phytohormone extraction and untargeted metabolomics, but we really, I don't know. That's a good, I mean, what we could do is we could use another set of species like Spidoptera frugiparida, which is another type of like caterpillar. And we could put that on the plant to check if it's kind of not just Spidoptera exigua. In this project, we did focus on Spidoptera exigua, but for future reference, that would probably be a good idea.
Okay, we should probably move on, and if you have more questions for Arianna, you can ask it during the break. Okay. I apologize for the technical issues. I think the problem is we're using Microsoft instead of a real computer here. And I'd like to introduce our second speaker, who's Brandon Williams, who's working in the Harrison lab together with Penelope Lindsay, and who will be talking about his work on transcriptional regulation and microbial interactions in medical practice.
Hi everyone, so my name is Brandon and I'm a student at the State University of New York at Brooklyn, and today I'll be presenting about examining the transcriptional regulators of corpuscular development and what I did this summer in the Harrison lab. So I just want to start off talking about farming. So farming is something done around the world and it's required to feed our growing populations, but unfortunately farming requires the mass spraying of chemical fertilizers to maintain crop productivity and crop output. And this is because key nutrients such as phosphorus are very limiting in the environment, and they're a finite resource, meaning they're not going to be in forever. And with phosphorus production projected to peak in 2033, we can't always expect to exploit this resource. And furthermore, when phosphorus and other chemical fertilizers are applied in high enough amounts, it can lead to runoff in adjacent marine environments, as seen in this photo. And plus, when plants don't get enough phosphorus, they turn into angry mangrove roots. So this is a plant known as Metacago concatula. It is a legume studied here in the Harrison lab, most importantly because it is able to form what is known as arbustule mycorrhizal symbiosis. So it interacts with this fungus called Glomerulus versiforme, and it can interact with the spores in its surrounding environments to form these structures known as arbuscules within its roots, where this nutrient exchange occurs. So after the fungus kind of penetrates the roots, it kind of goes down into the cortical cells, and it penetrates the cortical cell and kind of develops this branch-like structure known as an arbuscule. And it is here that you see this nutrient exchange between the plants and the fungus, where the plant provides the fungus with carbon in the form of lipids and carbohydrates, and in return, the fungus provides the plant with phosphorus, nitrogen, and other micronutrients. And this is important because if we can kind of figure out how to hone this nutrient capturing ability, we can kind of possibly decrease the use of fertilizers in the future or stop the use of fertilizers altogether. So I want to talk about the symbiosis. In order for the symbiosis to move forward, it has to be the signaling between the fungus and the plant. And that happens with this common symbiosis signaling pathway within the plant. So it starts off with the signal perception and transduction with the MYC receptors and other proteins. And here, like the plant is kind of translating the signals coming from the fungus before it kind of colonizes. And this leads to nuclear calcium spiking, which in turn kind of activates this interaction between DMI3 and Cyclops. And when this occurs, you see this activation of some important symbiosis genes, which is awesome because that allows the symbiosis to move forward, and you see the development of arbuscules. So the Harrison Lab studies the molecular mechanics behind arbuscular mycorrhizal symbiosis, and I wanted to kind of mention one of the important genes, Cyclops. Cyclops is a gene found in Lotus japonicus. This is another legume that is well documented in the literature, and it is also able to develop the symbiosis with fungus. But recently, it was discovered that there is a ortholog of this gene, or like another copy of this gene, in the species Medicago chincantula. But even more recently, it was discovered that this gene in Medicago has two copies of itself, IPD3A, IPD3B. And with more being known on what IPD3A does and how it interacts within the plant species, not much is known about IPD3B. And that is kind of what my project was exploring. So we had three primary objectives. The first, we wanted to kind of see, we wanted to study the interactions of IPD3B as compared to like IPD3A in its interactions using the yeast 2 hybrid method. Second, we wanted to figure out if root architecture was significantly different between the mutant genotypes and the wild type genotypes. And lastly, we wanted to figure out if phosphorus concentration or nutrient availability affected percent colonization within these different genotypes and the mutant as compared to the wild type. So our first experiment was the yeast 2 hybrid experiment. And with this experiment, we used yeast as a way to observe protein-protein interactions. And what this involves is we cloned our genes of interest, IPD3, and we transformed it into vectors containing either a DNA binding domain or a DNA activation domain, with the idea that when we make these strains of yeast together, after we transform them into compatible strains of yeast, after we make them, hopefully we'll see this interaction, and this interaction of proteins within the yeast will lead to this expression of a reporter gene, which shows up as blue. So after we made it the yeast, this is what it looks like. And as you see here, we have IPD3A and IPD3B, and we kind of wanted to see the interactions with DMI3 and some other important symbiotic transcription factors. So the first thing you can kind of see is that IPD3A has this important interaction, or has a strong interaction with DMI3. This is important because DMI3 is involved in calcium, kind of like decoding the calcium signals during arbuscular mycorrhizal symbiosis. And IPD3A is well documented as interacting with DMI3. So this was kind of our positive control. But interestingly, we see that IPD3B also interacts with DMI3. This is cool, but other than that, there was no other overlapping interactions between the two genes, as seen here. So just to recap, IPD3A and B both overlapped interaction with DMI3, but beyond that, there were no other interactions. So this seems to suggest that IPD3B may function differently than IPD3A. Our next experiment was the root architecture experiment. This lasted about 2 weeks, and this is kind of our setup of growing the plants. And after 2 weeks, we harvested the plants and kind of used a program known as ImageJ to measure the roots and kind of do like an analysis of root area and the amount of lateral roots they have. So as you see here, I kind of chose pictures that are representative of the 5 genotypes. You can see that R108 is highly branched and has a high root area, but if you compare that to IPD3B or IPD3 double mutant, there's a huge difference. IPD3B was interesting because it seems to resemble the IPD3 double mutant much more than it resembles IPD3A or the wild type genotypes. And our data reflects this. So with mean sum root length, IPD3B was not significantly different than IPD3 double mutant, but it was significantly different than the wild type and the 2 and IPD3A mutant. But when you look at mean number of lateral roots, IPD3B was not significantly different than IPD3A, but it was still not significantly different than IPD3 double mutant. And I believe that this experiment should be repeated to kind of confirm these results or see if this pattern reappears. So in summary, the root architecture experiment, root architecture did vary between the genotypes, and IPD3B resembled the double mutant much more than it did the other genotypes. And our last experiment was the phosphate experiment. With this experiment, we had our 5 genotypes of plants, and we had 3 groups. A high phosphate concentration, a low, and a medium. And after 3 weeks, we harvested and analyzed percent colonization to see if nutrient content kind of affected this, like how well colonized these roots were. So before we even analyzed the roots or harvested them and kind of cut the roots up, you can already see that high phosphate plants are much happier than the lower medium plants that are kind of like sad looking. So, like when we harvested the roots, we dyed them and analyzed them under a microscope to count the arbuscules. And with the low phosphate concentration, you can see some pretty arbuscules that come up really good. And if you look at the medium phosphate roots, there's not a difference between them. And what I mean by that, there's not a difference in morphology. Like the arbuscules look the same. But when you look at the high phosphate concentrations, you see some kind of funny looking roots as seen with IPD3A and IPD3 double mutants, where our arbuscules are kind of smaller and they just look kind of funky. So going to our percent colonization, we found that there was a negative correlation between colonization and phosphate concentration. As you increase phosphate concentration, the percent colonization decreased. So first, I want to draw attention to just how stunted colonization was within the IPD3 double mutant and IPD3B. And right now, there is no difference between IPD3B and IPD3A that we were kind of looking to see. But when you go to the medium phosphate, there is a bigger difference between IPD3B and IPD3A, kind of alluding to the fact that maybe they are functioning differently, like we believed before. And when you go to the high phosphate concentration, colonization drops to like really low levels, and the lines between the genotypes kind of blur. So the results show that IPD3, that morphology did kind of differ within the high phosphate IPD3B and IPD3 double mutant. Phosphate concentration did impact root colonization or percent colonization, and IPD3B seemed to resemble the double mutant much more than it did the other genotypes. So in summary, it appears that IPD3A and IPD3B are functioning differently, and this should be explored more so in the future because they're both important in allowing our vascular microbial symbiosis to go forward. And this was supported by our two experiments, the root architecture experiment and the phosphate experiment, where we measured our vascular colonization. And looking forward, I believe that the yeast 2 hybrid experiment should be repeated, which me and Penelope are in the works of doing right now. I believe that the root architecture experiment should be repeated as well to confirm our results. And lastly, me and Penelope are in the works of doing a yeast 1 hybrid experiment, where instead of looking at protein-protein interactions, we're looking at protein-DNA interactions, which is interesting. And I want to thank the Harrison Lab for fostering a family-like community and making it so fun this summer. I want to thank Penelope for pushing me to do the best I can and always being really positive. And I want to thank the Boyce Thompson Institute for having me here, the NSF for funding, and for Cornell for allowing me to stay on our campus for the summer. Thank you.
Any questions? Question?
I may have missed it, but why were your yeast named blue? Are you using some sort of a lactase thing?
Yeah, it's a lactase reporter thing. So when there's the interaction of the two proteins, when you make these, they activate the expression of that lactase reporter.
I'm just curious about what the interplay between the fungus and the plant's root system is. Like, does the IPD3 version, I guess, does it sort of act to like press defense mechanisms on the plant's part, or is it like encouraging some kind of like nutrient transfer? And like, if it is like a sort of repression of defense, is there any ideas like looking for a defense response, like in these?
I wouldn't say defense response, but it's like the signaling that's happening between the plant and the fungus. So like when these two interact, they're activating these other genes. So if these don't interact, or if IPD3 doesn't interact with with BMI3 or CPMK, there isn't this activation of genes, and the plant doesn't go forward with the development of our vascules. I'll try to answer your question.
Now I'm just curious, like what is like how the vascules are they're allowed to form.
Mm-hmm.
I'm just curious, I guess, like what are some of these symbiosis genes doing?
Yeah. So for example, there's the interpretation of these signals within the MYC receptors in BMI1 and 2 and other proteins. If they don't kind of translate these signals, the plant isn't going to be responsive to this fungus trying to colonize it. And some other mechanisms might come into play.
Yeah, so thanks for a great presentation. So I'm just curious, I realize there's some limits to the yeast 2 hybrid, but just to speculate a bit, do you think that there's necessarily some unique function of value in IPD3B, or is there perhaps just some sort of competition for binding there? And does anything in your other data kind of maybe suggest one way or the other?
I wouldn't say there's competition between IPD3B and A, because as you see in the IPD3A and B mutants, there's still the development of our vascules, meaning something's still happening there. I wouldn't say, like, we're not sure like exactly how IPD3B compares to IPD3 yet, and we, or the Harrison Lab, plans on kind of pinning up IPD3B up against some more interactors responsible or active in our vascular microbial symbiosis, and hopefully we can kind of see just what it's kind of interacting with altogether.
Okay. Sorry. Maybe you can ask during the break. We should probably move on and see moderately on stage. So our next speaker is Lily Lofton, who's working in the Swinger Lab with Xinma, and she'll be telling us about soft rot disease and potatoes. which I have to admit I know nothing about. So I'm here to learn.
Good morning, everyone. Thank you all for being here with me today. Again, my name is Lily Lofton, and I'm here from Delaware State University, working with the USDA ARS and the Emerging Pests and Pathogens Research Unit. The title of my talk today is The Screening of Wild Potato Tubers for Genetic Resistance to Soft Rot Disease Caused by Dickia dianthus. To begin, I'll be starting off with just a little bit of a background. The common cultivated potato, Solanum tuberosum, is a staple crop for many human populations around the globe. This crop throughout history has been clonally propagated, and this system makes it very good for quality genes to be reinforced, for example, the taste and the texture of tubers and their size. However, it has severely reduced the genetic diversity of the crop. Due to this makes them very susceptible to single pathogens. An emerging threat for the crop now is the bacteria Dickia dianthicola. One very important factor of this bacteria is that it has the ability to lay dormant within intercellular regions of the plant. That means that a plant can have the infection, however, it will experience latent symptom development. This is very important because this bacteria has infected the potatoes at the seed tuber level. These tubers are being produced, and when they are distributed naturally, then they are being propagated, and the plants are coming up with the infection already inside of them. As we are right now, we do not have any external measures to control the infections caused by Dickia dianthicola. Along with this, because of this, we concluded that internal resistance or genetic resistance would be the best way to combat this infection. However, the cultivated potato right now does not have any known complete resistance to Dickia dianthicola. Because of this, and because of the limited genetic diversity in cultivated potatoes, we have searched into wild potato relatives to find this genetic diversity and to find resistance. Another very important factor of our assay is that it is the broadest resistance screening of wild potato tubers for resistance to Dickia. Our objective of the essay is to conduct a coarse screening of wild potato tubers to find those with the highest resistance to Dickia dianthicola-induced soft rot. Here are some pictures of the symptoms caused by Dickia dianthicola infections. On the left here, we will see poor crop emergence. This would be the example that I said with that when the seed tubers are propagated, they're coming up already with the infection, so we have poor crop emergence. On the right, we have a tuber infected with soft rot. You can see here, just completely macerated tissue. You open it and it looks like pudding almost. In the next slide, on the left, we will see a collapsed plant in the field, and this is due to a collapsed vascular tissue of the plant. And if we were to zoom up right on the base of the plant here, we would see a black necrotic lesion, and this is called blackleg. This is another infection caused by Dickia dianthicola when it infects the stem of the potato plant. Moving on to our materials and methods. For this project, we received 392 wild potato accessions from John Vandenberg of the U.S. Potato Gene Bank in Wisconsin. And these wild potato accessions were collected from southern North America, Central America, and down through South America. Going through and inoculating and evaluating each 392 of these accessions would complete one experiment, and for these experiments, we completed 2 replications. For one experiment, we took 4 tubers from every accession, and 3 of these would be inoculated with our bacteria, and 1 would serve as the negative control. To inoculate the tubers, we took a flathead wooden dowel and punctured a hole in the tuber. We then injected them with 10 microliters of our bacterial suspension of Dickia dianthicola. This is a main-derived isolate. It's called ND2030. Following the injection, we then sealed them with petrogel, which is very similar to Vaseline, and this served to keep both the bacteria and the moisture inside of the potato. The negative controls were punctured in the same fashion, however, they did not receive any liquids, and then they were encased in a paraffin film to protect from contact contamination. All accessions were then incubated overnight at 30 degrees Celsius in moist chambers, and the following day, they were all cut open at the site of the wound, and the widest part of the lesion was measured. So here's a picture of the evaluation. I'm going to have another picture, make it a little easier to see. You can see all of the lesions in the top of the tuber. and then one more. This is a close-up. So again, we measured the lesions at the site of the widest site of the lesion. Moving on to our results. So throughout the experiment, we noticed a lot of variation among these tubers. This variation was seen in the size, the color, the flesh color, and the skin color of the tubers, as well as the size and colors of the produced lesions. Here are 3 histograms that we produced for every experiment. On the top, we have experiment 1, experiment 2, experiment 3. And these histograms represent the frequency of lesion size occurrence. So if we were to take 1 bar from the histogram as an example, 1 bar would represent 1 lesion size. Then the height of the bar would represent how often this lesion size occurred. As we can see, if we look at these, from every experiment, it is consistent that the majority of the lesions occurred between the 4 to 8 millimeter size range. And then it tapered down on the left and the right for the low and high extremes. Our target right here, however, is the red and blue regions for the lowest lesions because we are looking for resistance. The red region, far to the left, represents the lowest 1% of the lesion sizes. The blue represents the lowest 2 to 5% of the lesion sizes. All the way to the right, the yellow represents the highest 95%, and the blue, the highest 99 to 100% of the lesion sizes. Following the histograms, we produced these graphs for every accession. Now, these accessions, well, the graph is still respective to the colors that we saw in the histograms. So if we take one of these, it is one accession, and the graph shows how often this accession occurred in the categories that we chose. So how often this accession occurred in the 1% or 2 to 5% categories of the lesion sizes. Just sectioning out a few individual graphs here, if you follow me to the bottom right, we see a species of Solanum tuberosum, subspecies indigenum, that occurred very frequently in the largest lesion category. Now this could tell us that this is a very susceptible accession. On the bottom right, we have Solanum jamesii and Solanum boliviense that occurred in both the smallest and the largest categories. This could tell us that these 2 species have a large variation in the size of the lesions. Now, if we go to the top here, which is actually our target, we have the top 3 accessions that prove to be the most frequent in the lowest lesion sizes. Now, one thing very interesting about all of these top 3 accessions is that they were all of the same species, and this is Solanum microdontum. So Solanum microdontum proved to have the highest frequency of the lowest lesion sizes. And from this find alone is telling us that this species has the highest resistance to Dipia denticola infections. And here's a picture of the Solanum microdontum. You can see that it has very little symptom development at the site of the wound if you're comparing the inoculated tubers with the negative control. Very little. Here's another picture of another one of the microdontum. And now I'm going to compare this to a very susceptible accession, just so you can see. So now here we have a susceptible accession, and you can see an increase in the macerated tissue and the lesion size. Very, very good. So we can go back to that. Just resistant to a very susceptible accession. So our key points and next steps for our experiment. Key points is that genetic variation and resistance diversity exists among the wild potato accessions. And then our most important find for this was that Solanum microdontum occurs with the highest resistance to Dipia denticola out of all of the species and accessions that we tested. Our next steps is to conduct a secondary screening of the Solanum microdontum species. And now, instead of doing coarse screening, we'll be going into a finer screening, and we can limit it to what specific plants have the highest resistance to this disease and infection out of all of them. Along with this, we will be collaborating with Amy Czarkowski of Colorado State University, and we will be trying to figure out if tuber resistance directly correlates with stem and plant resistance. As of right now, we do not know the answer to this. Our long-term goal is to incorporate this resistance found among the wild accessions into the cultivated potato genome to give the cultivated potato resistance and more protection against this bacteria. And I would like to thank the USDA, ARS for their funding and means of this work and this research. Brian Swingle and Shing Mom, who are actually present in our audience today, my supervisor and mentor for everything that they have taught me and helped me, and healthy to learn and to grow, and then Shing again for his data analysis. John Bamberg of the University of Wisconsin and U.S. Potato Gene Bank for collecting and giving us these live potato accessions. Keith Perry of Cornell University for providing us with commercial potatoes to initially work with our experiment and figure out the correct ways for this to work. And Walter DeJong, also of Cornell University, for consulting with us about potato breeding and data analysis. Thank you.
Questions?
Yes.
Where was the micro adopted from? Where did you get it from?
It was actually shown to be in Colombia and Bolivia. So we have South America on the west coast of South America from that region. It wasn't one specific country, but yes.
Do you know if your group, or I guess your collaborators have genotype or resequencing data for the microdontin, and in terms of future work, there might be an opportunity for associating the genetics of the microdontin to particular resistance?
I believe that's a goal of ours. I don't think that has been completed yet, but yes, finding the mechanisms for this resistance and producing the sequences for these for easy inclusion would be a goal of ours, but it has not. Yes.
Your experiments were all done in this incubation, right, overnight.
Yes.
How does this, it's kind of a somewhat artificial system, right? How does it relate to actual potatoes grown in soil? Did you test it, or are you going to? Or?
So that's a very great point, because we're producing these environments in the lab, but that differs greatly from the field. So all of these experiments are going to have to be replicated in the field to get the proper results. But in the lab, we were trying to recreate an environment for the bacteria to grow so we could see the real resistance of the species. But like you also said, they will have to be replicated in the field to see how they actually interact. And that was the other goal of ours, seeing if the tuber resistance correlates with the stem and plant resistance. So if we find a resistant tuber, this might not mean that we have a resistant plant. So that's a very important thing to continue with the research to figure out. Yes?
I'm just wondering how the bacteria is inflated in nature. Do you expect that the resistant phenotype to be found using the transmission vector?
Okay. Well, the bacteria actually, it can transmit, it can transfer through some insects, and it can also stay in some of the water and soil. And it enters plants through stomata and blooms, and there it enters through the intercellular regions where it actually degrades the plant cells in there. So to control contamination, just because it does spread these ways, we would have to completely separate infected from non-infected tubers, because it can transmit very easily and just transfer. Does that answer your question? Okay, great. Yes.
Thank you. Sorry, I probably should. So our next speaker is Patrick Mendoza, who's been working in my Stanley together with Phil Cochran.
Okay, well, thank you for the introduction, Dr. Jander. Like Dr. Jander mentioned, I'm going to be talking about leaf polarity, and particularly within the domesticated tomato plant, or Solanum lycopersicum. So for a brief overview of the topics that I'm going to cover, I'm going to have a brief introduction, background, what is leaf polarity, what background experiments have been conducted so far, how the Scanlon lab continued those experiments, and what the findings were, as well as what potential next steps there are available. So as far as leaf polarity, it's important to know that leaves grow along 3 axes. Now, the first is the proximal distal, and that one measures how far the leaf grows outward from the main stem. The second one is the medial lateral, which measures the distance from the mid-vein to the lateral edge. And lastly, but more importantly for this discussion, is the adaxial-abaxial side, or the axis of the leaf, which does determine the leaf polarity. And basically, it determines the top and the bottom of the leaf. And it's the opposing forces of the adaxial and the abaxial against each other that actually produces the medial lateral, or the flattening of the leaf. So why is this important? So it's important for a couple of reasons. And the first is that the flattening of the leaf does help to maximize photosynthesis. You have a maximum coverage area, and with the adaxial at the top and the bottom side of the leaf, you can orient certain organs that at the top of the leaf that would particularly capture more sunlight, maximize photosynthesis. And on the abaxial side of the leaf, you could perhaps have more stomata, you could maximize your gas exchange there. So it's important for many reasons. As far as the background experiments that were conducted so far, back in the 50s, Dr. Ian Sussex conducted some experiments on leaf polarity, and he hypothesized that there was a signal that developed from the apical meristem to the leaf primordia that kind of developed this polarity. And what he did was he conducted a microincision that separated those two portions to see what would happen. And the incision, you can see is right here. When he did that, there were some radially symmetric leaves, or conical leaves, that ended up developed completely of a baxillized tissue, or made up of completely the underside of the leaf. In addition to that, normal leaves developed on non-surgical cells, but there wasn't a 100% ratio of radially symmetric when this was conducted. So there was a mix of conical and non-conical leaves developed. So how did the Scanlon lab continue these experiments as far as leaf polarity? Well, so the idea was to find the genetic underpinnings that drove leaf polarity. And as a starting point, the Sussex experiments were used to develop these radialized or completely baxillized leaves. And you can see that with the arrow that's pointing down at one right there. Once that was complete, then tissue was collected from those radialized leaves, as well as leaves that develop naturally. An RNA seq was conducted on it. And without going into too much detail on RNA-seq, I'd like to say that once that was done, we were able to determine which genes were highly expressed in the conical leaves and which genes were much less expressed in those leaves by comparing the conical to the non-conical. Next steps in that was that looking at those genes that were highly expressed versus not so highly expressed, they were candidate selected based on for CRISPR constructs to see what happened when there were certain mutations on those genes. And there was one for the upregulated or highly expressed in the conical leaves, and that was the LRR gene. And there was one for the less expressed gene in the conical leaves, and that was the MY2 gene. The plants were received back, and they were also observed for possible phenotypic changes. So right off, there are a couple things that were noted. On the left-hand side, you have a typical wild-type tomato leaf. Now, there was an LRR, or highly expressed gene that was in the conical leaf that came back that was much more dense. It had smaller leaves, it was more compact. And the MY2 gene that came back, mutant that came back, ended up with an elongated stem. It had smaller leaves, it was more spread out, and it had a wider canopy. So what was done next? Well, the leaves were analyzed under a scanning electron microscope to see if there was any differences between, at least under 50 times magnification, between these types of plants. And you can see that under the wild type, that the abaxial, or the lower side of the leaf, is covered with trichomes or hairs that are very long and prolific. Now, on the adaxial side, or the upper side of the leaf, you'll notice that it still has those trichomes, but they are much shorter, perhaps not as dense. Now, the LRR leaf that came back still had the prolific trichomes on the underside of the leaf, and on the upper side of the leaf, there were actually very few. So there was some type of mutation that occurred there. Now, the MY2, you'll notice that both the adaxial and the adaxial have similar trichome structures, and that they actually look very similar to the adaxial side of the wild type. So there appears to be some type of polarity mutations occurring where you end up with potentially two top sides of the leaf as far as trichome development is concerned. Now, under a higher magnification where we can look at the cellular epidermis structure of leaves, we looked at the wild type plant. And you'll notice that the abaxial, or the bottom side of the leaf, has a jigsaw-shaped paving cell structure. On the adaxial side, they are much more rounded, a little bit more loose. Now, when you look at the LRR mutant, it still has that jigsaw-shaped pattern on the bottom side of the leaf. And it does also have the rounded, looser structure on the adaxial side. MY2 seem to follow the same pattern as the trichome structure, where you'll notice that both the abaxial and the adaxial are somewhat similar, and they're very different from, at least as far as the abaxial on the wild type, where it looks like the top and the bottom are very similar. So this is very promising to look at. And what we wanted to do was then consider next steps. So the next steps for this were that the mutant plants that we had received had all been mutated only on one chromosome. So they were heterozygous. So the goal is to then self these plants and see if there's a way that we can determine or find a homozygous mutation. So that involves harvesting seeds, planting them, and seeing if there is the next generation at a smaller proportion that has some type of phenotypic variation. Also, what was done was the continued genotyping of the first generation plants through cloning and PCR amplification. So, when conducting scientific experiments, there's surprises. Good surprises, bad surprises. So what we found was that the phenotypic plant, we do have heterozygous. And I lied a little before when I said there was one gene. There were two genes targeted in this one, MY2 and glutaredoxin. Both were heterozygous in the phenotypic plant. And as we received more and more mutant plants back, we found some that were non-phenotypic that tested negative for the heterozygous state. Now, as more came in and more sequencing was conducted, we got a surprise. And there was a non-phenotypic plant that displayed the same heterozygous cuts that were in the phenotypic. So, not ideal. But it doesn't end there. So we did also find the same results in the LRR plant as well. So the conclusion here is that there are two possibilities. The first is that there is human error in the sequencing of these genes. The second is that there was an off-target CRISPR cut that was actually responsible for these phenotypes. Now, we're leaning more towards the off-target cut because we did sequence in genotype, sequencing clone modes. So that's what we're looking at. However, since all of those plants were heterozygous, we still bred them, and we were hoping to find a mutant. And we did. So we found a plant that in the next generation LRR that had terminated meristem, it had ceased growth. And instead of having a dissected leaf or compound as in the wild type, it developed simple lobed leaves. And here is a close-up of that. So this was very exciting. And based on the information, we thought that there was a definite possibility that this was a homozygous mutant for LRR. However, we're still doing sequencing yesterday. We got sequencing results back, and it is possible that this is due to a combination of the cut in LRR as well as another off-target. So it's a little disappointing. But as far as next steps, we'll continue to monitor the T1 plant mutations, and we can look at these plants under, histology, under microscopy, to see if there's anything that's different in these ones that we might want to look at more. Now, because there were off-target cuts, if we wanted to find out what was responsible for the phenotypes, we would either have to do an analysis that determined which probable genes could have been the off-target cuts, or sequence the entire genome, both of which are a little daunting, and we'll see where that leads. Lastly, genotyping the T1 generation would still be in effect, and we have found some homozygous mutations, which was on the previous slide for one of the genes, but we'll continue to monitor for the other ones as well. So lastly, I'd like to give thanks to the generous donors, as well as BTI and Cornell for facilitating this research, the entire Scanlon laboratory, and in particular, my mentor, Phil Conklin, who took a horticulture student with basically little to no molecular or genetic knowledge, and gave him a very good crash course in molecular theory and methods of the lab. So, any questions? Yes.
So in terms of those plants that were genetically heterozygous mutants, but displayed a wild type phenotype, right? Are there known sort of like, I don't know if antagonists is the right word, but are there genes that I think with the MATE2? Or yeah, MATE2. So are there known sort of genes that like may sort of, there's like a balance there, or maybe your off-target cut is like a gene that the loss of this off-target gene sort of recompensates for any effects that the MATE2 genes alone?
That's possible. These genes were selected because they were previously hadn't been reviewed in the data. So looking at ones which might have compensated for that would be an analysis that could be conducted. Yes.
What is the gain function of LRR and MATE2?
So that's a really good question, and I might be wrong on this. But LRR it stands for leucine-rich repeat. And as far as I understand, leucine is involved in protein-to-protein communications, but all of them are involved in plant development, plant growth. MI2, I believe, is involved in meiosis as well as growth.
So I have a question also.
Yes.
Did you check the genome to see if there are likely off-target sites that might have been cut by this person?
No. So that we would have to do an analysis on the likely cuts for those and then amplify those genes. Yes.
Did you look at still model density and whether or not very neurons?
That's a really good question. And we could, because we do have SEM images of the phenotypic plants. So we could take those SEMs and load them into a computer program that would then count the number of stomata. We don't have that right now. Yes, that is something that would be really interesting.
Sorry, I think we need to continue. So our final speaker before the break is Lauren Hamm. She's working in Stefano and Lily's lab with Stefano himself, and she's going to be telling us about the editosome.
Thank you for that introduction. Like you said, my name is Lauren Hamm. Hi everyone, I am from Rhodes College, and today I'm going to be talking about using the ABI3 promoter to complement and subsequently rescue lethal mutations in genes vital to the editosome mechanism. Now, the term editosome refers to the overall process involved in cytidine to uridine base modification RNA editing in plants, which is quite a mouthful. So to start, I would like to explain what exactly is RNA editing. Now, RNA editing is a generally corrected process that post-transcriptionally alters RNA sequences to fix deleterious mutations in the genome. This process is particularly widespread across the overall phylogenetic tree of life. It can be characterized in organisms like viruses, Drosophila, trypanosomes, squid, plants, and yes, even us. Evolutionarily speaking, RNA editing had many unrelated points of convergent evolution over time. One of the most interesting being the evolution of cytidine to uridine editing in plants. Now, the general theory states that this type of RNA editing originated shortly after terrestrialization of plants into harsh and arid environments. Heightened exposure to harmful UV rays drastically increased the amount of harmful T2C mutations, which accumulated over time until aggressive mutation reinstating primitive deaminase activity allowed the plants a way to be able to convert properly encoded cytidines into uridines. Now, this process often results in some restoration of an amino acid or start or stop codon. A good example of this would be what you see here, the chloroplast indie ATP transcript, which is involved in ATP synthesis coupled electron transport, which is really, really important to the plant. Now, without CU editing, the start codon would never even be translated correctly, and you wouldn't see any type of translation for this entire protein. Now, the long-term goal of research into this mechanism is to be able to recreate it in vitro. And once we have succeeded in this endeavor, the editosome could be used to modify any type of RNA, including human RNA. Now, right now, there are many detrimental T2C mutations that we know about involved specifically with humans that can lead to anything like coronary spasm, lung cancer, or mitochondrial encephalomyopathy. All different symptoms that could be theoretically relieved through the harnessing of the editosome mechanism. However, this is still quite a long ways away due to the overall need for more data on this mechanism and the processes involved with it. What we do know, however, is the overall shape of the editosome as well as the different types of families involved in this process. Now, editing specificity is first initiated by the association of a member of the transfactor family into tripeptide repeat proteins, or PPR proteins for short. The PPR protein interacts with a related cis element that occurs immediately before an editing site. This protein then recruits either one or two RNA interacting proteins, which are thought to then recruit one organelle RNA recognition motif protein, ORMs, or an organelle zinc finger protein as well, OZs. Now, not much is actually known about the functionality of these last two families, but knockdown assays do suggest their overall necessity for this editosomal process. And altogether, these four major families make up what we like to call the editosome. Now, generally, when researching a gene suspected to be involved in the editosome mechanism, like the four families that I just talked about, we employ either knockout or knockdown techniques using loss of function of insertional TDA mutants or virus-induced gene silencing, respectively. However, there is a class of genes that cannot be accessed through these techniques because they are essential genes, meaning that mutants can't be recovered at the homozygous state, and they have low levels of expression, meaning that silencing is particularly difficult to achieve with these genes, which gets rid of both options, and you can see why that would be a problem. But for these specific genes, I've helped develop a new workaround method that utilizes the temporal expression of the ABI3 gene to help us come up with a new way to look at these genes. Now, ABI3 is a transcription factor that's located downstream of the ABA pathway, meaning it's responsible for the activation of many different seed-specific genes. By attaching the ABI3 promoter to a copy of the wild-type gene of interest, we are able to induce conditional expression of this gene during all stages of embryogenesis, where it's essentially vital for this gene to be expressed, while also allowing for the dissolution of expression after this developmental period is finished for the plant. In other words, expression of this construct allows for the survival of homozygous mutants that would otherwise remain inaccessible to the lab. But what's really, really exciting about this process is that it's not specific to the RNA editosome mutations, and you can actually use this process for any type of gene that falls into the category of being essential and having low levels of expression. So we've essentially created a pipeline where you can use this to fix a multitude of problems. Now, for my specific study, I focused on 5 genes, all of which fall into that category of essential genes with low levels of expression. Those being OZ2, GRRPB4, MEF8 and MEF8S, and DYW2. Some interesting things I want to know about a couple of these genes are that MEF8 and MEF8S, the PPR protein pair, are actually pretty well studied and aren't lethal in their own. But the problem is we can't study the interaction of these two genes because the double mutant is lethal. So I will be using these and transforming our construct for both of them into the same sets of plants. And the DYW down here at the bottom is acting as a positive control for the experiment. There's already been some success using ABI3 to rescue DYW2 in another lab that posted this about a week ago. And this will allow me to compare the completed process to previous work. That way we have a little bit of context for the overall situation. Also, for brevity's sake, at this point, I'm only going to be using OZ2 as an example to explain this process. That way we can get through things a little bit quicker and get to the brief that everyone is really excited about. Now, after isolating the gene of interest, OZ2, the first step is to transform it into a vector called the entry clone. This process is relatively simple. The gene is first amplified, and then single deoxyadenosine protrusions are added to the 3' end of the PCR product, which creates a sort of puzzle piece template, which you can use to easily insert it into the entry vector using a T over hangs that you can see in this picture. Now, I specifically use a tap amplification with a particularly low error rate called superfly green, but just to make sure that this process was completed correctly, I used PCR to determine the constructs were the correct size for what we were looking for, and then subsequently sequence the vector to make sure that there are no mistakes in the overall transgene. Now, once you have it in the entry clone can then be used as a base point from which the transgene can be transferred over into a destination vector, thus creating an expression clone. This process is really similar to the last step in that there are essentially sequence bookends called ATT01 and ATT02 that surround this transgene region and correspond to bookend sequences in the destination vector soon to be expression clone. These bookends are specific to the transgene region and allow for the transfer of a particularly large construct, which helps us keep the efficiency rather well. And just like in the last step, guess what? I double-checked this transformation using both PCR to ensure the correct size, and then sequenced the transgene a second time to make sure that there were no mistakes during this part of the transition. And the finalized construct should look something like this. Key things that I would like you guys to note in the transgene region, which is located between these two borders in red, would be the appropriate antibiotic resistance gene coupled with the ADA3 promoter and gene of interest complex. And that's really all you need for this construct to be successful in producing homozygous human progeny. I then transformed that finished construct into agrobacterium using a process called agroporation, which is, electroporation, which is once again double-checked using both PCR and genetic sequencing. Now, making sure that there are no mistakes in the construct is especially important at this process or the step in the process, because these agrobacterium will then be used as the delivery vessel for the transformation into homozygous or heterozygous mutant progeny. And putting these into the Arabidopsis mother plants makes up the last segment, floral dipping. But before we get to that, I mentioned earlier that homozygous mutants carrying both copies of the TDNA insertion don't really survive. But heterozygous plants containing just one copy of the TDNA insertion do. So the first step in this transformation process begins with identifying which plants are homozygous wild type and which plants are heterozygous. To do this, I completed rather extensive genotyping using 2 rounds of PCR, the first with primers specific to the wild type allele, and the second with primers that are specific to the mutant allele. And when only the wild type section exhibits amplification, as you can see here, we know that this plant is homozygous wild type and not very useful to us in the lab. But if you can see both the wild type and mutant sections being amplified, then we know that this is a heterozygous plant, and it can then be used as the mother plant for floral living. Now, these mother plants are allowed to flower, and I then submerge the structure, the floral structure, into a sucrose mixture that facilitates transfer from agrobacterium. The plants are allowed to recover from this particularly invasive step, and then are allowed to produce the seeds for us to use. But despite being conceptually simple, the step actually takes quite a bit of time, because after you go through this dipping process, they have to recover, grow seeds, the seeds have to be harvested and then planted, and even after you do all of that, you then have to sequence the mutants to make sure that it's the homozygous progeny that you're actually looking for. But at the end, it should be well worth it. And as of right now, I have completed this entire process with OZ2 and prepared the construct for both the GRRP4 and DYW2 with the methane-methane-S pair, particularly close behind. Now, since I've already completed the bulk of the work developing a stable construct with these first 3 steps, the lab, after I leave, should be able to finish this process for all 5 within the next 1 to 2 weeks if they continue at my current rate of work. I mean, it's going well so far. And then after we've got all this figured out, we just have to wait for seedlings to grow. We should get the homozygous progeny that we're looking for. Once we get them and everything is all wrapped up, the newly produced homozygous mutants will be used to collect data on each of those 5 mutations. Mutant phenotypes like dwarfism, which you can see here, or something like respiratory deficiency, can help explain which processes are affected by a specific mutant, as well as how widespread the overall damage is. I would like to include that this process of comparison isn't always particularly clear-cut, but even despite that, any information that we can get from a mutant phenotype would be helpful regardless. My lab will also be determining which sites have been affected, as well as how they have been affected, by providing us insight into the organoid transcripts involved and how it interacts with other protein families and other editing sites. And data like this can be used to help further our understanding of RNA editing and puts us one step closer to being able to master this process in vitro. So I would like to take a little bit of time here to thank everyone involved in the sponsorship of this project, as well as the overall Hands Lab, who was incredibly welcoming into their tight-knit group. It was a wonderful experience. I would like to thank Dr. Stéphane Bentelila, who actually couldn't be here today because he's enjoying his time in France, but he's still here in my heart. I would like to thank Andrew as well, who's actually here acting as a proxy mentor as he has for the entire time that Stefan has been gone. I also like to take some time, because no one's done it so far, to thank Delaney for being super helpful and for being a mother hen for the group and keeping us all in line. And if you can wait a couple minutes before we go and take a break, I'd be happy to answer any of your questions.
No, go ahead.
You should go ahead. He's in my lab.
So I'm a little familiar with what a gene knockout is, but can you explain what a gene knockdown is?
Knockdowning is we're using silencing, essentially. So the knockout is that we're putting these tRNA inserts that essentially take out the function of the protein itself. That way it just doesn't work. But with silencing, we're not actually removing the gene itself, we're just removing its efficiency, the way that it works. So the gene isn't gone, so it's not immune, but it still is immune in a sense. I'm not doing a good job explaining this. Does that work? It's like an in-between method. Okay. Yeah.
So it's like some of these editing genes you can knock them out and the plants are living, but like these ones, they're not. So like, is there any idea about like, you know, what gene, or like what transcript is it they're editing that's so important for that early stage?
Actually, we're not entirely sure, other than the basic families that we're in. We've figured out what families these are supposed to be in based off of similarities in the motif structure of the actual sequence itself. But because there's been, besides F8, and a little bit of UIW2, there's been almost no research on any of these, so we don't actually know what they're involved with or how. All we know is that if they don't have them, the plant dies because it can't edit something that's particularly important in the survival of the plant.
Maybe time for one more question.
Yeah, you wanna? Yeah.
So I probably missed this, but what exactly is the editosome kind of like a protein that sits on RNA usually, or?
Yeah, it's a post-transcriptional process, so it is on the RNA sequence itself. The editosome is actually a conglomeration of multiple trans factors or proteins that interact with each other to go in after the fact and fix problems or mutations in the genome that otherwise couldn't be fixed, and change it to what it needs to so the proper encoded product works. So it is just a conglomeration of a bunch of different proteins that are actually different at different sites. So we do see a high amount of specificity with PPR proteins and a good mix of all the different kinds. So the example I gave you was chloroplastic instead of mitochondrial, which is what OZ1 is involved with, but in some of the main examples that we have for mitochondrial, we don't see OZs present. There's a lot of mixing and matching, but it's just kind of like a little group party with the RNA sequence.
So let's take a 15-minute break and try to be back here at 10:45 for comments.
I love the sass. Oh God, I died. A few zingers.